everyone, my name is Heather Gorham, Principal Investor at Flying Fish Ventures, and this is Applied Context. In this episode, I flew across the pond and sat down with Simon Kohl, CEO and co-founder of Latent Labs. Previously, Simon was a research scientist on the AlphaFold team at Google DeepMind and was co-author on the AlphaFold 2 paper that was published in Nature in 2021. It is no exaggeration to say that this team's contributions have been some of the most significant in science and technology in the 21st century. In fact, so much so that the team's lead, John Jumper, and DeepMind CEO and co-founder, Demis Hassabis, were both awarded the Nobel Prize in Chemistry this year for this amazing contribution. Simon and I take it back to basics, going through the model's architecture, his time on the team, as well as some of the stuff he's working on now. I'm so excited for you to join us on this discussion. Let's dive in. Thank you for being willing to be a part of this. I'm excited to be here in London.
Yeah. Thanks for having me.
You've been a part of a very important team that delivered one of the most important contributions, as many say, not only in machine learning, but to modern science in the 21st century. This team is obviously AlphaFold. It's been a while since that has been published, so I'm excited to talk about some stuff that's happened since then, but would love to hear, maybe before your time at DeepMind, highlights where you kind of were and then how you ended up on the team.
My background really actually is in physics. So I studied physics at KIT in Karlsruhe in Germany up until my master's, where I worked in high energy particle physics. And essentially, we had lots of data from a particle collider experiment in Japan, and you really required machine learning to sift through that data, make sense of that data. That's how I came across machine learning. And then I guess the bug bit me. I wanted to do more in that area. Decided to actually do a PhD in machine learning, but really was looking for an application that would sort of ground me in a domain that I would be, excited about for the course of the PhD. And I found that in medical image analysis. So I moved to Heidelberg in Germany to do my PhD at the German Cancer Research Center, working with real patients, MRI and CT scans, and essentially trying to build AI systems that work similar to radiologists, how they would interpret these images. And pretty early on in my PhD, I realized that these images are often ambiguous, so you want to arguably model a distribution over how you could interpret them, because that's how the radiologists actually in some way do it. Like if you ask 10 radiologists, you get 10 often slightly different answers, sometimes wildly different answers. So there seems to be this distributional nature to the problem. Pretty early built alternative models for that. And it so happened that one of my works was retweeted by Ian Goodfellow, and that sort of led to DeepMind seeing some of my work. Fast forward a couple of, maybe a year, that turned into an internship at DeepMind. And so I moved to London to intern there in the DeepMind Health team, working with Olaf Ronneberger, the inventor of the UNet. And that work was successful as well, resulted in a spotlight presentation at NeurIPS. And essentially they wanted to continue in that path, in that direction, and offered me to do my PhD, finish my PhD there while being a full-time research scientist. So I jumped on that. It was quite a nice opportunity to have. That also went well. I was able to finish my PhD there. And in hindsight, sort of serendipitously, there was like a slightly abrupt change that coincided with the end of my PhD, in that Alphabet as a company realized they had several different health teams, and they were going to pool them in what became Google Health. That meant the DeepMind Health team largely moved over to Google, and I, alongside a couple of others, stayed at DeepMind, and I was looking around which teams to join. With my physics background, I felt the protein structure prediction problem was interesting and related at least to molecules. So had that bridge, and I decided, why not join that team? And that was at the end of AlphaFold 1. So people were already writing up sort of the Nature paper for that, and I had some time to ramp up in the domain, because the last time I really had anything to do with biology was in high school. And so I had a couple of months to ramp up, and then I was there from the start for the development of AlphaFold 2.
Wow. So yeah, talk about like totally being thrown into a different domain, and I think that's a really unique thing about DeepMind. I don't know if everyone's familiar with how their applied teams are set up, but you get paired, right? It's like they're interdisciplinary teams of machine learning engineers and scientists, and also people of the domain. So you can kind of learn very quickly with like the people next to you, right?
Yeah, that's right. Basically, we were a small team of interdisciplinary scientists, like you said. I think, perhaps we were just a little over 10 people at the start of AlphaFold 2, maybe just about 10. Actually everyone, was a computer scientist of sorts, but with some, bio background, some of them. And it so happens that I think often you find physicists in these areas as well, including myself, some other physicists on the team. Perhaps that describes well the makeup, but you're right, definitely different backgrounds, but everyone, more than I think sort of literate in ML, definitely. But down to the domain scientist is, I guess, what I'm saying.
Yeah. So you end up on this team. There's a push to make it to one of this competition that we'll talk about in a second. But not everyone, I think, is familiar with proteins. So very quick, high-level overview for people who are outside of the field. How should they think of proteins?
Yeah, I think there's a pretty simple view that you can evoke there, which, proteins are these molecules that are made up of a chain of amino acids. You can maybe picture them as like literally beads on a chain, and they're like one connected to the other. And each of these beads can take on 20 different types. Those are the 20 different amino acids. And this sort of chain of beads, by the laws of biochemistry and physics, folds up in space and takes on a shape, a 3D structure. And that's really what a protein is in many ways. Like from a computer scientist's perspective, you could look at this as a sequence made up from letters with a 20-letter alphabet. And of course, these beads aren't actually beads. They're like themselves small little molecules, if you will, that have a sort of atomic substructure made of different atoms. And really sort of proteins, zooming out a little bit to their significance, they are the building blocks of life, and sort of every life form on the planet. Sounds a bit grand if you put it like that, but it is true, are made up of these. You and I are, and they are these like little nanomachines, if you will, that perform the essential functions of our bodies, of life, really. And maybe just to slightly add to that for people to make the connection, our genomes and our genes are essentially the blueprints that code for proteins. So there's this pretty much, in biology there's always exceptions, but pretty much one-to-one mapping from a genomic sequence to a protein sequence and its structure. And so, yeah, all the proteins that are in our bodies are coded for in our genomes.
So they're very important. They're in kind of everything living. And these chains, loose chains, are unstable. And so to get to stability, they fold, right? Is that the right way to trying to get to, why protein folding and understanding that was so important?
Yeah, I think the way I would put it is sort of the space of possible amino acids is actually vast, and that point has been made a lot, right? If you think about it, like if you have sort of, let's say, n of them, and you have 20 options for the amino acid type, then you have 20 to the n possibilities, and that quickly becomes huge space. And I think the thing to say about this space is that most of this space is probably not actually folding into a valid structure. So there's only a subsection there that can actually take on a sort of shape, and sort of zooming out a bit more and adding a bit of color, the sort of structure and shape of these proteins lends them their function. At least mostly that's true. And And so, yeah, the significance is that in the life sciences, whether that's sort of basic research or maybe biopharma, there's a lot of interest in understanding the functions of certain proteins, how they interact with other proteins, and particularly, maybe understanding when these interactions that are natural break down. That's, in a sort of simplified view, actually what disease often is like breakdown of a certain pathway that involves protein interactions. And so that's really where this drive comes from. Like for a long time, we've been trying to understand protein structures, and in the past, this has been done via experiments. And there's nothing wrong with experiments. The trouble only is they take a long time. Very often it's taken perhaps one PhD, so like 3 to 5 years or something like that, to like resolve one structure. Sometimes it wasn't even possible at all for various reasons. And so it's just a painstaking process to do it via experiments. And if we were able to, which often we now are with tools like AlphaFold, if we were able to predict the structure just with a forward call of a model, and that structure was pretty accurate, that'd be awesome. And that was the idea.
Yeah. So at the time, you guys were heading towards this this competition, which is like the Olympics of protein folding, right? Which is the critical assessment of protein structure prediction, CASP. And AlphaFold 1 had done it in 2018 and had the highest score kind of ever seen before in the competition. Yeah, what was the process in getting there?
No, yeah, you're exactly right. CASP is this sort of great competition that has been actually running for, I forgot when it started now, but I think 3 decades by now. And that shows the long-standing interest in it, and it actually also sort of brought all of the relevant groups in these areas from all over the world together every second year, and sort of it is this sort of blind assessment, which means experimentalists that determine new structures, they actually were holding out certain structures from being published so that they could be sort of a really sort of blind held out test set for this competition. So none of the entrants knew what they were going to be tested on. And that's, I think, huge credit to the organizers who set up a fantastic competition and have done that over such a long span of time. And really that enabled quite a clear picture of what worked and what didn't. And like you said, for a long time, things, it wasn't like they didn't work. It was just like not to the extent that anyone would have wanted to pick a model. It was sort of specialized groups working on that. I think that's probably fair to say. And then, like you said, AlphaFold 1 came around, and it was better than anything before in the sort of 60 GDT range. And you described it correctly. It's sort of this metric that assesses how good your prediction of the structure for a sequence maps onto this held out true experimental structure. And yeah, you can sort of loosely think of it as an accuracy, like you said, 100% would be perfect. And so 60, is decent, but no biologist would have sort of used AlphaFold and not done an experiment, or really sort of perhaps made any critical sort of derivations from these structures that a costly experiment depended on, or something like that, right? Like just you wouldn't trust a 60 GDT prediction enough. But then, yeah, our goal was to, when we started AlphaFold 2, to lift that to as high as possible, of course. And going from 60 to something like 90 felt, when we started, like kind of impossible, right? You were looking back on a field of like that has worked on this for more than 20 years, and also AlphaFold 1, right? Like great team working on this, like also took significant time and efforts, and great ideas went into it, but that quote unquote only reached 60. So it felt like, oh my gosh, how are we gonna push this to 90 or above? And so, yeah, I think one of the really interesting things that we've done when starting the project was actually tossing out AlphaFold 1. I think that took a lot of courage and belief that something entirely else was going to work. And in many ways, that paid off really well. And I think we had good reasons to believe that. I mean, we kept pushing on the AlphaFold 1 type architecture, and it just wouldn't, go up a lot. Like, we could get it a bit better than maybe it had entered CASP at that time, but not like, not like significantly better. So like, okay, we need something entirely new. And yeah, I think that was really well executed by the team and sort of took that courage, I guess. Let's see, yeah, I think, of course we had a lot of the understanding of how to do research in this space. So there's like a lot of bleed from what had been learned by AlphaFold 1 that went into AlphaFold 2. So it wasn't like, we tossed everything out, but like some of the ideas, I guess the key sort of modeling ideas in AlphaFold 1 was actually treat proteins as sort of a distance representation and run a convolutional net on that. Of course, much more, but in a sort of, if you like squint at it, that's what it was, like a convnet running on sort of pairwise distance representations. And so we sort of tossed that out, at least the convolutional bit, made it all sort of such that any position could attend to any position. So formulating that with attention mechanisms. Basically, a lot of sort of, I think, biological and physical insight went into crafting the architecture from there. So happy to expand if you want me to. But
yeah, no, I think that would be a great way to go into the architecture of AlphaFold.
Yeah, maybe sort of connecting to where I was coming from just now. Essentially, what's so great about deep learning in many ways, I think, is that you can build in notions and prior knowledge about your problem in flexible ways. So it's not like you're hard coding certain knowledge. You can if you want to, but you can do it in sort of softer ways. And I think that's some of the power of deep learning, that you nudge sort of the model into the right direction of the solution space by the way that you formulate the architecture, but it's still flexible enough so it can actually really sort of ingest the data and learn from it in the flexible ways that you would want. Really, I think what we did was we built on sort of this prior knowledge that the field had arrived at, which is that co-evolutionary signal across sequences can tell you something about what's in proximity in the structure. And really the idea is quite interesting and profound in a way. So what you do basically, given a sequence that you want to predict the structure for, you search for evolutionarily related sequences. Once you have a stack of these, you look at what co-mutates, what correlates across different positions in that sequence. And essentially from that signal you can deduce a whole lot about the structure. For instance, positions that are close, they more often have to co-mutate, otherwise they would perhaps break a bond or introduce a charge where it shouldn't be charged. Similarly, like conserved parts, they may correspond to the core of the proteins where you don't have a lot of flexibility to introduce mutations, things like that. So in a sort of perhaps, you could maybe say that this evolutionary signal is like a shadow of the structure. It tells you something about the structure. It doesn't pin it down exactly, but it gives you constraints.
If the shape was changing all the time across these different sequences, the function, it would no longer be serving the same function. So you can kind of deduce from these similarities.
Evolution certainly does select for a function, but what is really selected for, it's difficult to say. And certainly, you could sort of sequences that align, that have quite a bit of a different function, or take on a different structure as well. So the network has to be smarter than just figuring out these correlations. I think that's part of what AlphaFold added. There's many things more, but you can't sort of just go with these co-evolutionary signals. Like the model, I believe, also has the ability to ignore some of them when they don't fit the rest of the pattern, things like that, or irrelevant sequences could also be ignored, and so on. So it is quote unquote smarter than that. Yeah. But yes, that's the sort of core idea.
And so that block is the multiple sequence alignment, right? In terms of kind of the pre-processing stage.
Yes, yes. So basically, what you want to do is you want to extract the juice out of this multiple sequence alignment. And the way we've done that is this architectural block that we call EvoFormer, and where Evo stands for evolution. And essentially, we sort of embed the MSA with attention type modules, so not unlike transformers really. And then we interleave that. That was one of the innovations in AlphaFold 2 with a pairwise representation. And the two representations, the representation of the sequence derived from the MSA, and the pairwise representation encapsulating all the pairwise residue interactions, they interact. And there's a lot of detail to that, which I could speak to, but the sort of core idea is, you want to find out in the sequence which pieces, which residues, which amino acids, I use these terms interchangeably, they largely are, which interact with each other. And for that, we do this interleaving between the pair stack and the single stack, as we called them. That's EvoFormer. And that's sort of supervised with different losses. So it being a deep learning system, you formulate your goal or task as a differentiable loss. And we had what's called a BERT loss on the sequence. I don't think it's terribly important, but it did help a little bit, where you sort of mask out certain parts of the input sequences and try to paint them back in. And then we had another loss on the pair stack that supervised the distance prediction, essentially, so that the pair stack part was incentivized to infer these distance relationships. And that's the EvoFormer block, which in and of itself doesn't result yet in a 3D structure. So then what comes next is block we call IPA, or it is made up of IPA, invariant point attention blocks. It's a sort of, imagine it, think of it as a 3D transformer operating on sort of frames in space, where every residue can be characterized by a translation and a rotation. That's what we mean by a frame, and this transformer is built such that it can update the translations and rotations of these frames, one for each residue. And essentially, we then use the embeddings found by EvoFormer to predict the explicit 3D structure, including the sidechain. So again, there's a lot of detail that you could zoom in on, but essentially, in many ways, EvoFormer gives you sequence and pairwise representations, which we then translate into a concrete 3D representations of where the atoms are in space with the next block. And then, perhaps also worth mentioning is the confidence estimates that AlphaFold comes with. That's actually something that I've personally worked on a lot, and I came up with what's called PLDDT, the predicted LDDT, and that's now a confidence metric that people use a lot outside, actually, both to understand when a structure prediction is reliable and where, but also in protein design, which we may speak about later, I imagine. People use it a lot to select and rank designs, and sort of the reason why it's become so popular and impactful is that it's actually well calibrated. So, that just means, you can usually trust it.
Yeah, I wanted to make sure we got to talk about this, because this was something that you got to directly contribute on. Why is understanding how confident the model is on locally important when you're looking at these structures?
Like, it's worth in general, right, to know whether your prediction is good. Like, globally is as important in a way. But then even on the local level, it's very interesting, because perhaps, certain section of your prediction are less good, and you would hope thereby they are predicted with a lower confidence. And you would want to know about that. And why that's interesting is because perhaps what you really care about knowing a lot is the part that is confidently predicted. So then you wouldn't mind, right? It's fine that the other end of the protein isn't as confidently predicted if what you care about is confidently predicted. And similarly, where maybe that hasn't quite materialized, but sometimes can get it, is often people are interested in sort of what's the word? Essentially in making changes to the sequence, like a point mutation, for instance, so changing one amino acid type, like how does AlphaFold react to that? And I mean, the honest truth about that is it often doesn't quite capture it. We actually sort of openly disincentivize people from using AlphaFold that way because it's not necessarily built to do that. It's a sort of function of how much it actually uses the MSA as well, where you do have a lot of mutations. So it, in some ways, is built in a mutation-insensitive way, but sometimes it can get it, and that can be reflected locally in the PLDET, but it's not really what it's built for.
You published, and this is also what you took to CASP14 back in 2020. It was the highest score ever seen, a score of 90, considered like a solution level, dramatically better than anything else. What did it like, just being on the team and the reception from the community, what did that feel like?
Yeah, it was really cool, of course. It was fantastic. I mean, it's probably worth saying that, I guess seeing it internally before you tell anyone else is one thing. Actually, there were sometimes scares where we're like, oh my gosh, now we've like surpassed on our internal metrics 85 GDT. Like, is that even, like, how can that be so good? And like, we sometimes had these scares, like, did we train on this test protein and so on. So there was that. And then basically, as the way CASP works is it's actually a competition that goes on over several weeks, and they release a certain number of structures per week. And as this unfolds, they also release the true experimental structures, so you can start to see. I think it's beyond. It's when it's done, but they start releasing some, and so you get a sense. You can obviously do the maths yourself. Like, how good were you on these like initial ones? And the very first one, I believe, was our worst, and we're like, oh, this is like 80 or something like that. Obviously before that would have been awesome, but internally we were seeing much higher numbers. So then we got this sort of first protein structure released from CASP, and we're like, oh my gosh. Maybe we weren't quite as good as we thought, but then from there went uphill. There was that element. And then, the external reactions, once the result was actually made public, was tremendous, was really good. I think people liked that over time we, open sourced it, made it available. John Jumper, the team lead, had a talk at CASP where he went into some depth of the architecture, and I think overall that was all well received. And I think the really cool thing for us was seeing researchers basically use it on actual problems, in actual biological problems, and also reconfirming with sort of experiments after CASP that AlphaFold's correct. And that was like a really exciting thing for us to see. The other thing that was unexpected to us was a bit later, once we open sourced the model. People actually used it immediately, literally within the first 1 or 2 weeks, to do things we didn't think AlphaFold could do. Like, maybe now you would call that prompt engineering. But very quickly people found, for instance, that it was often able to correctly predict the structure of 2 proteins in complex. So what I hadn't mentioned is, AlphaFold 2 was built for single protein structures or single chain proteins, and it was not trained or built to predict how 2 structures would jointly fold. And actually, often it was possible to do that with some simple tricks, some hacking, some prompt engineering. And we were surprised by that. And that's sort of, to some extent, the sort of wisdom of the crowds, right? Like you open source something, and then like people use it in ways, we thought weren't going to work, and they proved us wrong, which was really exciting to see.
Part of what you mentioned in the other applications and kind of what else this unlocks, you mentioned protein design. I think there's, and maybe you could talk a little bit about this. I know that you spent a lot of time in that. How does being able to, have technologies like this, just the loop is much faster, change our ability to design proteins? And for maybe those that don't know kind of what novel or de novo proteins are, I think that'd be great to hear as well.
Yeah, no, that sounds great. Yeah, like you mentioned, I worked on protein design as well quite a bit, like after AlphaFold. I started a project on that, a team really, and ended up co-leading that as well. That was still at DeepMind too, and at Latent Labs now we're broadly speaking on working on protein design as well, certainly on engineering biological systems, and so yeah, I've worked in that area for a couple of years now. Yeah, I do think AlphaFold had a profound impact on that field. Essentially, people are interested for perhaps obvious reasons, but we can also go into that in more depth, but are interested in designing proteins with new functions that could be research tools, could be diagnostic tools, could be therapeutics, all of these reasons. And really, I think the protein design field has made a huge leap over the perhaps last 5 years or so, from I think only very small proteins perhaps being able to be redesigned, very simple proteins. There was like some early successes, but it was very limited, and certainly the success rates were very low, and that's in part because there wasn't a good way to know before you tested it what the chances of success were. All of that, there's a lot of detail which we could go into, certainly on some protein families that would have worked better than on others, and there were early successes, and the field developed a very good understanding already how to design proteins, but I do think there has been a leap coming from ML models and from AlphaFold in particular as well, both in terms of how people can now rank designs computationally before they test them, but also in how machine learning-based design systems are built. They often borrow ideas and notions from AlphaFold, really. And then, perhaps it's worth saying that it is still the case, however, that in sort of protein design, nothing trumps experiment at this point in time. Like, you have to go test it. It's not like AlphaFold is 100% reliable, not at all. That's not what I'm saying. You can use it to rank it. It often gets it right, but we're not in a point in time where you design a protein in silico and you can then expect it to definitely work.
And so there's some areas that we understand less, which is maybe how proteins interact with each other, these protein dynamics and things like that. Are there like open research questions within some of these things that we can't yet do that are particularly interesting to you?
Yeah, I think I find it very exciting to think about what could this field look like? Like, why can't we do what I just said, right? Like, why can't we design something computationally at the moment and it will essentially just work? It's definitely true that this is an incredibly complicated problem. Like, if you think about what these designed proteins or proteins period are doing, right? They're like floating around in a cell, interacting with lots of other molecules. They usually have a certain function, meaning they interact with a particular recipient of their message, if you want to put it that way. And a lot can go wrong in sort of this sort of cellular environment. It's not even contained to that, right? Proteins get outside of the cells. They are messengers between cells. It's like fantastically complex, but so were other areas of science before we found perhaps the right way of describing them and thinking about them. Like, if you think about celestial mechanics or perhaps even at the sort of atom and electron level, before we perhaps had the description language that now is quantum mechanics, like, we couldn't make any predictions in that realm. And I think we're sort of at a time where I think we're finding sort of, or at least one good way of dealing with that chaotic system that is biology, which I think, as others have pointed out, might be machine learning, and that's what I find very exciting. So can we come to a point where we can specify the function of a protein, and it's usually the hard thing is it's not just one function. You want it to fulfill many functions all at once, and that's just the perhaps the attributes of one molecule in this sort of entire system. Like, what if we could perhaps even simulate that system and understand sort of at a perturbational level, like what happens if we introduce a new molecule or take one out, and then beyond that, can we maybe design the whole system? What could we do with a technology like that? And that's the sort of that level of control in biology. I think we don't have right now, but it's what we're working on, what we think will be possible, and I think it's extremely exciting.
Is that like looking at things more at the atomic level, like because really kind of at any material, whether it's protein or anything else, atomically, I mean, if we're able to understand what's happening at a better level there, does that kind of unlock this more universal engine?
I think this is all developing, but what seems clear is the sort of constraints of the area in terms of what type of data you can collect, and how perhaps you can collect more data in certain areas and so forth. So I think you have to sort of work with what's feasible, and clearly there's a whole lot of structural data. There's a whole lot of structural models right now where you can model interactions at that level. But there's probably orders of magnitudes more on the sequence side, because it's just easier to obtain data for that, whether that's sort of protein sequences or perhaps even sequences of entire genomes, different types of functional assays that characterize functional changes under perturbations. And I think just like looking at the constraints coming from that and what you can do with that, I think it's clear that we need multimodal models that co-integrate these different data domains, and I think that also does make a lot of sense. So that's at least the conviction I hold, that you want to sort of bridge these different scales and actually jointly cross-integrate information from these different scales.
So you've since left DeepMind. You're now working on Latent Labs. Do you want to talk at all about kind of what you're working on at least at a high level?
Yeah, we haven't said a lot, you're right, which, at a very high level, we are working on making biology programmable. We are building large foundation models based on generative AI systems trained on large data sets. We have a fantastic team, so other people from DeepMind have joined, including other people from the AlphaFold team. We also have a fantastic team on the biology side, with people joining from other biotech companies that are well known. We're really bringing the two areas together, allowing us to swiftly test what we're engineering and designing, feeding that back to the model, collecting training data. We have two sites. We're actually here in London for the foundation models team, and then our second site is in San Francisco, where we have a wet lab and a computational bio team. That's probably as much as I can say before we come out of stealth.
No, it's great. This has been wonderful. I think it was great to hear kind of the structure, and obviously there's been so much done in the field. There'd be no way we could cover it in 40 minutes with what AlphaFold unlocked. But I think as more attention in the world is coming to AI, it's good to kind of understand that this, how far we have come in such a short amount of time. I think across many different modalities, we've seen this exponential improvement in systems. Obviously, people are very familiar with what's happening in language models. I think it's equally important to highlight some of the other fields which have progressed really quickly and have great impact. I think it's one of, it's where I like to spend a lot of time. It's some of the best use of machine learning is in fields that directly help a lot of people, the planet, and like two very important things. So it was great to be able to talk through.
Likewise, no, thanks a lot for having me on this.
Thank you so much for watching this episode of Applied Context. If you enjoyed this episode, please consider subscribing to the series so you can be notified of future episodes coming out. I'll also leave some links in the video description so you can see what Simon is up to now, how you can get a hold of me, and I hope to have you around for the next episode. Thanks so much. See you then.